Hello, my name is Mason Dana, and I'm presenting the research paper Augmented Want Signaling in a Mammalian Model of Accelerated Aging. So the background and introduction for this paper shows that resident and circulating stem cells and progenitor cells are critical for ongoing tissue maintenance and repair. Stem and progenitor cell depletion or dysfunction might contribute to aging, but the contribution of these cells and their dysfunction and how it relates to aging is still incompletely understood. And so they're going to examine how stem cell dynamics in a genetic model of accelerated aging and how that works together. So they're going to be looking at mice lacking clotho-protein expression, and they're going to call them clotho mice. And these mice have a shortened lifespan and exhibit a number of early onset age-related changes, like arteriosclerosis, decreased fertility, and skin atrophy. So clotho is a transmembrane protein that is also secreted outside of the cell. So it's generally believed that secreted clotho is the form that most likely is mediating the protein's longevity effects. So essentially, the clotho that ends up leaving the cell is probably the clotho that is causing mice and other mammals to live longer and age less quickly. So these researchers in a cell culture model, they explored the want-clotho interaction that resulted in a suppression of want. And so want proteins regulate cell-to-cell interaction. So essentially, they wanted to see the clotho and want interactions in a cell system. So here are their results. So here's an altered stem and progenitor cells in clotho mice. And A is showing the LRCs within the skin follicles of 80-day-old wild-type and clotho mice, and that's on the top of A, the two first panels. And then a higher magnification underneath them is showing the representative bulge regions that have been stained for CD34. B shows TA cells identified by positive brown nuclear BRDU staining after skin wounding. And so C, right next to it, shows an assessment of skin closure 4 days after creating a 1 centimeter wound in the mice. So essentially, they created a wound and wanted to see if the closure rates were different for the wild type and the clotho mice. So D shows evidence for senescence within the hair follicles of clotho animals as assessed by B-Gal staining. And it shows the foci of GH2AX and 53 BP1 in red, and the 4' and 6' DAPI stained nuclei in blue. And so the percentage of positive nuclear staining in either wild type or clotho follicles is shown with a standard deviation there. So E, panel E, shows an SAB-Gal staining of the small intestine in both the wild type and clotho mice. F shows a determination of the absolute number of the C-KIT and SCA1 in bilateral femur and tibias of the wild type and clotho mice. And G shows a representative cell cycle analysis of HSCs from the wild type and clotho animals, demonstrating a decrease in HSC sequence and an increase in proliferation. And DNA content is also displayed there. So in figure 2, it shows the interaction of clotho with want and inhibition of want signaling. So A shows the HEK293 cells are transiently transferred with MyCAD hemagglutinin, HA, tagged expression constructs encoding WANT3 and murine clotho, so just CLO. And a reciprocal co-immunoprecipitation demonstrates the two extracellular Klotho repeats, KL1 and 2, followed by a single pass transmembrane domain flagged with full-length Klotho and truncation mutants were assessed for WANT binding. Sorry, and that's a Western blot in A. So B shows at the top a schematic diagram of the Klotho demonstrating the two repeats. I'm sorry that I'm repeating myself here. Shows the two repeats in the genes, and then there's a single pass transmembrane, and then the actual Western blot underneath shows the WANT binding of these different passes. So C shows an HEK293 cells are transferred with a WANT3 expression construct and the amount of full-length Klotho expression vector, along with either an active or inactive WANT luciferase reporter. D shows a conditioned medium mixes that were incubated for 24 hours with the HEK293 cells that were previously transferred in C. And all of the WANT activity measured shows a ratio of TOP and FOP activity that was obtained from a single experiment performed in triplicate and are representative of at least 3 similar experiments. So figure 3 shows inhibition of WANT signaling by Klotho in vivo. So that means in an actual mouse. So A shows endogenous WANT activity in skin from 14-day-old wild-type and Klotho mice crossed with Top-Gal reporter strain. So essentially, they just want to see like where WANT activity is being expressed within the mouse cells themselves. So B is showing WANT activity in tibias of 14-day-old Klotho Top-Gal and wild-type Top-Gal mix. So these are crossbred. So it's a Klotho / Top-Gal mice and a wild-type Top-Gal mice. So I guess you're seeing the interaction between Klotho and Top-Gal there. So C is a longitudinal microcomputerized CT section of the tibia of a 3-month-old Klotho and wild type. So the top is a Klotho and the wild type is at the bottom. And there you can see that the Klotho mice has a lot more of that white sort of material within the bone, whereas the wild type it's pretty bare. So D shows a horizontal 3-dimensional reconstruction of a cordial bone and a trabecular bone. So the cordial bone is in yellow, the trabecular bone is in green. And so again, we see the same pattern, whereas the Klotho mouse bone is a little more filled in there than the wild type. So panel E shows a calculation of the trabecular and total bone volume from the tibia of 3 pairs of 3-month-old wild type and Klotho animals. So they're essentially looking at the density of the bones here. And so along with what we saw in panel C and D, E is also showing us that these wild type bones are less dense, less filled in than these Klotho bones. F shows a quantification of the percentage of B-Gal positive follicles from 12-day-old Top-Gal mice that were injected 4 days earlier with a control virus and an endovirus encoding the WANT inhibitor, or another endovirus that encoded Klotho. So they chose about 500 random follicles, and they assessed those for the condition. So they're looking for LAC-Z positive follicles and the percentage of that. And so here we see, so the first column is just the control virus that they used. And there we see that a large, so almost 50% of the follicles that they chose, so about 250 follicles, were positive for LAC-Z. And if you look at the second column, that's the virus that had the WANT inhibitor, and we see that it's less than 10% of the follicles are showing this LAC-Z, and then in the Klotho, slightly more, but I don't think that's a very large statistical difference there. So G is showing the hyperplastic skin phenotype of K5-RT-TA, TAT-WANT1, and those are transgenic mice that were treated with doxycycline, which is blocked by prior injection of an endovirus encoding for either DKK1 or Klotho. And so the mice were killed and observed 21 days after birth. So figure 4 shows senescence induced by increased WANT activity. So A shows BRDU incorporation of MEFs grown in the presence or absence of WANT3A conditioned media. And cells were assayed from passage 1, so P1 there, through passage 4, P4. So B shows senescence associated with B-Gal staining, SA-B-Gal, for MEFs, and that's in P4, grown in standard media or mixed with the indicated L cell conditioned medium. C, panel C, shows MEFs in P4 with or without continuous WANT3A exposure. They were assessed for senescence associated heterochromatin, HP1A, and then and then the activated DNA damage response. So D shows the level of BRDU incorporation in P4 for MEF cells continually treated with the WANT3A media, and either vector transfected or Klotho transfected conditioned media. And so there we see that the Klotho transfected media, you see more BRDU positive cells. So panel E shows the growth of MEFs in standard media or supplemented with WANT3A at different concentrations. So we see WANT3A at 10 nanograms per milliliter or 30 nanograms per milliliter. And P7 cells were stained for activation of H2AX. So panel A, the last panel, shows skin sections obtained from those 5K-RTTA TET-WANT1 transgenic mice that they had in the beginning of this. And they were either treated or untreated with doxycycline from postnatal day 2 through 21. So at day 2, they started treating them with the doxycycline. And the senescence-associated marker GH2AX is induced in the setting of continuous vivo WANT1 exposure. So in this paper, they actually do demonstrate that Klotho acts as a WANT antagonist, and that chronic WANT stimulation might contribute to stem cell depletion and therefore aging. So this shows a connection between aging and the WANT signaling pathway, which is pretty cool, I think. So I think in the future, you could possibly target this WANT signaling pathway and potentially cure or combat aging and age-related diseases. But yeah, I hope you enjoyed this. Here are my references. Yeah, have a great day. Bye.